is from Birgit from Wageningen, and Birgit will talk about GWAS for feed intake and methane emissions.
Yeah, hello everyone. Thanks for the introduction. So I'm going to tell you a little bit what we did in Work Package 4 with feed intake and methane emission. I would like to start the presentation with thanking all of my co-authors, and you see there are quite a lot of names on the slide from the different partners who provided data and carried out the GWAS analysis. So this was really a nice collaboration here, and I would like to mention that we were able, so not only within BOVREC, but we also were able to get additional partners outside BOVREC who had feed intake and methane emission data from the Global Dry Matter Initiative available. So this was really nice to give us a little bit of a boost in in terms of number of animals included in the analysis. So small introduction. We have heard, yesterday Christa gave a really nice overview about the aims of BOVREC, and well, the overall aim is to provide a catalog of functionally active genomic features in cattle. And BOVREC key traits that we should focus on were biological and environmental efficiency. So where we focused on feed efficiency and methane emission. So I think methane emission was not in the project from the beginning, but as it turned out, I mean, and it's really an important topic, as it turned out that the partners in Work Package 4 have access to methane data, we just thought, okay, we will have a go and include methane emission as well. Yeah, but as you probably know, I mean, these traits are new traits. They're really difficult to record and expensive to record. So large-scale recording is still an issue here for methane emission and feed intake, meaning that the size of the reference population for GWAS analysis and genomic prediction is limited, also, of course, then leading to limited accuracy. So the objective here was then, okay, can we gain accuracy if we join forces and combine the data sets across countries to identify relevant genome regions related to both trait groups. And here for feed efficiency, we focused on dry matter intake, and for methane, it was methane production that was measured in grams per day. And as the speakers before myself, so we have carried out a meta-GWAS across dairy and beef cattle populations to see if we can gain information from the different populations. Good, so I'd like to start here to give a short description of the different data sets included. So we had access to dairy data sets and beef. So for beef, there were two populations included, a very large data set from University of Alberta in Canada, where it was Angus, Charolais, and the composite breed, Kinsella composite. And then there was a smaller data set from FBN, where it was a Holstein Charolais cross. And then we have a group of dairy populations where it was all Holstein populations except from Luke in Finland. There it was a red Finnish breed. So the phenotypes recorded in the different populations. So for DMI, all recorded phenotypes were based on, so the traits were coming from measured in feed bins where scales were included. So there were different kind of animals recorded. It was dairy cows, so in beef cattle there were also heifers and steers included. And then we had the dairy and beef cross, which were bulls, I think. And for the dairy part, we have to mention, so we did the recording was at different lactation stages. So for some populations, we really had access to recordings across lactations, but some populations only had recordings for a specific time period, few weeks of the experiments. Then for methane, It was even a bit more challenging here. So there are 3 different recording techniques used in the different populations. The one is the green feed. So you can see this one on the, I've put the pictures of the different measurements techniques below here. So it's on the right-hand side, that's the green feed, which is considered the golden standard. Then the sniffer, which is installed into automatic milking system, and that's on the left-hand side. And then we have the SF6 technique in the middle. And here as well, we had different lactation stages of the cows where the phenotypes were recorded. Good. So I'll start off with describing a bit, the meta-GWAS workflow. So each of the partners carried out their own local GWAS analysis, and we agreed on a joint imputation pipeline. I think that was similar also for the work that has been done in beef and also for the mastitis work that was developed by Hubert Pausch from Switzerland. And all partners basically followed this imputation pipeline, maybe differing using, not all using the same software, but following the general principle. And I've brought a table here to show you the number of animals and imputed sequence variants for both trait groups. As you can see, for DMI, the population size in the different from the different populations, it varies quite a lot. So we have from 140 animals included in the GWAS from FBN, for example, up to 7,500 in the Canadian data set. So it was really, so with rather small data sets. When we look at the methane production, so it's even smaller. At that time, there was really very few data sets available with very small number of cows recorded. So it was the largest data set at that time was provided by Spain, and all the others were so between 300 and 400, 500 animals available. So in total for DMI we had 13,104 animals, and for the methane it was a bit more than 3,000 animals. So I cannot really compete here with the numbers used in the speakers before from Marie and Gautam. However, we have to point out here that this is really the largest data set available globally. So we thought it's still worth to really have a go and see if we can find anything. Good. So each partner carried out, I call it a local GWAS on their own data set. So they used the GCTA software. You can see the model here. So where the allele substitution effect with B is included, and we also fitted a polygenic effect U. The phenotypes used in this model were either deregressed breeding value, it depended a bit what was available, and other partners just corrected the phenotype for fixed effects and used this then as a phenotype in the GWAS analysis. And then all this information, the results of the local GWAS, was then sent to us in Wageningen, and then we carried out then the meta-GWAS. We have used the software Metal that has been described before a little bit already. The standard error method, genomic control was included. Before running the analysis, so we have standardized the variance effects by the genetic standard deviation. We have included variance with effect size within the standard deviation of plus minus 5 standard deviations, and a minor allele frequency of 0.005 was applied. And then I have run 3 different scenarios, which are called all, Holstein, and beef. So in all, it's really we have included all data, all populations that are available. And then we did an analysis where we used only the Holstein data sets, and then an analysis for beef. And for beef, it was mainly the 2 populations, one from Canada and one from FBN. Good. Then, in addition, my colleague Annick Baumann at Wageningen, she also carried out a gene-based analysis. We have used the GCTA software with the FastPath method implemented there. So there it's, the principle is that you aggregate the effects of variants within a specific gene. So we have used the default settings here, so where a region of 50 kb plus minus, the gene was included. So we had LD calculation for LD pruning was based on the 1,000 bull genomes dataset run 7. We have used the Bonferroni corrected significant level that you can see here, and in the table below you can see the number of genes tested per trait and scenario. So overall, of the 2 traits and also the different scenarios, it was about 26,000 genes that were included in the analysis. Good. So then I'll start with the results from the meta-GWAS for dry matter intake, where we used all data sets available. And you can see the Manhattan plot here. So again, this was based on 13,000 animals and about 25 million or 25.6 million variants. And you can immediately see, so there is a nice peak on chromosome 6, and there are other peaks then on chromosome 14 and 20. So this was really nice result. So what we see here was already found by our Canadian colleagues as well in a previous publication, where they have only, of course, the data set that we had was part of the data set in this publication. So what we found here was, confirmation what was already published. Then, as I said, we did analysis using Holstein populations and beef populations only. And then, when using Holstein only, so the picture is a little different. We had close to 5,000 animals here included, close to 19.5 variants. And as you can see from the Manhattan plot, so there were no clear signals. However, there are maybe some promising regions, but no clear QTL signs. And for the beef, this is a confirmation of what we have seen before. So this is driven by the data set from Canada. We also checked whether the QTLs were present in the FBN data set. However, it's really, so these signals are these QTLs are really present only in this Canadian data set. Good. Then the results from the gene-based analysis, and I have here the one for Holstein and the beef only. So for Holstein, there were some significant genes on chromosome 10. However, when looking then at the variants, so they are counted only for a very small proportion of the genetic variants. And for beef, you can see it on the left-hand side graph, Manhattan plot. This, of course, lined up very nicely with the Manhattan plot from that I've just shown before. Good. Now, coming to the results that I would like to present for methane emission. This is the Manhattan plot for using all populations available, based on this little bit more than 3,000 animals and 22.6 million variants. And here we don't see any clear peaks. However, it's also not really flat, but so there are maybe some regions to explore further. However, no clear peak was found. We also checked whether there is a difference between Holstein and beef. And you can see from the Manhattan plot, so also in beef alone, which was in this case actually the Canadian data set only, so there were no clear signals for methane production. And then the results for the gene-based analysis for methane for Holstein and beef, it was, yeah, you can see from the Manhattan plot, so there were only few significant genes. In Holstein, it was on chromosome 2, it was a long non-coding RNA, and in beef, it was the SMOX gene. However, we couldn't validate in each vice versa in the population, so it was really only present in beef or in Holstein. Yeah, and other than that, as you can see, so we didn't find any significant genes. Good, so to sum up, so, well, we knew kind of before, so dry matter intake and methane emissions are a complex trait and probably very difficult. What we have seen for dry matter intake, so we confirmed the QTLs that are already published in the beef, so we couldn't see this in dairy breeds. However, we have passed this information on to a work package 7 for genomic prediction and to see whether there is potential benefit there in terms of accuracy of genomic prediction. And Renzo and Marzi will show the results later on. For methane emission, we didn't see any clear candidates in this data set so far. However, I really want to point out here, so it was really a challenge. It was really a heterogeneous data set across countries, so in terms of the trait definition for DMI and methane emission, and so there are different recording techniques. Just to see whether there is an effect of the recording technique, we had also carried out then just grouping populations with the same, using the same recording technique. However, this also did not give any, did not show any clearer signals as compared when using all data sets. Okay, and with this, well, I'd like to acknowledge the funding and also the WUR consortium partners. It was a really nice collaboration. Thank you very much.
Thank you very much, Birgit. Are there any questions for Birgit? So.
Hi, Chris, CRV. Did you find any crossover between the putative locations because of the high correlation between...
Sorry, can you say again, Chris?
Did you find any effect in those putative locations for the methane? Did any of them have similar signals within the dry matter intake, given the strong correlation between feed efficiency and methane?
No. Actually, the correlation between methane and DMI, so that's the strong correlation that you're referring to, so that's actually not confirmed yet. So in Wageningen, we are getting rather low correlations at the moment, so it's 0.2 to 3. So I know there are others. So I think this correlation still has to be investigated.
Yeah, but coming into the same direction, have you checked, because I've seen this peak on chromosome 10, is there an overlap between the two for DMI and methane?
No, it's not the same. No.
Okay.
So we have a question online. How did you combine the methane emission phenotypes obtained by the different methods, laser, sniffer, green feed?
Yeah, as I said, so we really, in this all scenario, we just put them together. All, so just assuming as being the same trait. But of course we know, well, now 2 years later, we actually know the genetic correlation between these methods, yeah? So for example, between the green feed and the sniffer, we get a correlation of 0.7. It's not bad as a proxy. However, you would consider it as different, genetically being different traits.
Okay.
What I did try was to, as I said, combine the sniffer populations only. So there were 3 populations using sniffers. But even then, well, the knowledge that we have now, so for example, the sniffer in the Netherlands, the sniffer phenotype is different from the sniffer phenotype in Spain.
Mm-hmm. Okay.
So.
Okay, thank you. Okay, yes.
With regards responses in terms of what should our priorities be in cattle breeding. The methane was the most ambiguous. And it's interesting that your results are also ambiguous. But also there is obviously much interest in, is there some way which breeding can seriously reduce methane emissions? So from a point of view of someone who is not an expert in genetics, so at this stage you have seen no particular results, but you have given very clearly that you are taking a very small amount of data. Is it realistic to expect a lot more data to be able to be a bit more precise, or is it?
It is. Well, actually, at the moment, so I can give you examples that I'm aware of. So in the Netherlands, we have now 8,000 cows phenotyped for methane emissions. In Denmark, it's close to 10,000 cows, if I'm correct. So it's really worldwide, the recording of methane emission is increasing. So there, and actually in Wageningen, we have just started to coordinate a global program called Global Methane Genetics to bring initiatives together to join forces, kind of similar what we tried already, but just using more data, how to effectively or more efficiently use data for genomic prediction. And this program also has funding to really increase the number of phenotypes on a global level. So we are, for example, we are aiming to have 50,000 cows phenotyped. So this is definitely underway.
Thank you.
Okay. Thank you very much, Birgit.
Thank you.
Let's move to the last presentation. Sorry. Excuse.
Sorry, Birgit. Maybe a little bit of a provocative question. Isn't there the danger that when we breed for reduced methane emission that we end up with pigs?
Yeah, that's a good question, and that's of course, we are all very, it's a really hot topic, and we are all going for methane emission, but of course we need to take into account the so we still need to want a healthy cow, and need to take into account, still keep the cow as a cow and keep her healthy.
Okay.